and also the invitation. It's a real pleasure to be here at Mount Sinai, which is one of the powerhouses on exposome research, I would say, and I'm really valuing the collaboration with the Institute. I'm currently the cluster lead of the European Human Exposome Network, which I will introduce later. And so what I wanted to do today is basically give a little bit a European context of exposome research, what is happening basically within that network, and then go a little bit later into one of the projects that I lead within that network to give a little bit more detail. So if we think about the European context, I mean, that's not different than what we have heard here today, and that is, of course, that there's full recognition that the environment is an important driver of health. And this is the picture that Doug Walker already showed with the population attributable fraction of the genetic component of disease, which you can see basically varies between a few percent to about 40 to 50% if we think about asthma. It's also basically estimated that if we think about environmental pollution, that contributes to about 20% of the deaths in the WHO region of Europe. So there's full recognition that the environment plays an important role in the health of European citizens. That's also basically recognized within the policy briefs and policies that we have. So the major challenges that has been laid out for the 21st century within Europe is basically summarized by the 3 Cs, which basically stands for climate, and that's climate change, chemicals, and HBM is one of those programs that really focuses on that aspect, and it's basically cities or urban health and all the pollution and things that come with that. That is also basically formalized in what is now the European Green Deal, which basically calls for a strategy to protect citizens' health in Europe and well-being from pollution and environmental deterioration. So that's basically loss of biodiversity from different sources of exposure. And 2 programs that are really relevant, I think, for exposome research, and I will come back to that later, is the Farm to Fork strategy, which basically is talking about sustainable food production, healthy food, and reduction in the use of pesticides. And is the zero pollution strategy, where the right of the European citizen is basically to grow up and live in a zero pollution environment. And what I specify here are the current missions that the European Union is developing, which focuses on cancer, healthy ocean, climate neutral cities, and healthy soil and food. That this is not just basically paper that is pushed out by the European Union is basically, I think, exemplified by the fact that Ursula von der Leyen, the current president of the European Commission, basically spoke about the zero pollution ambition, both at the time that she actually accepted her post as the president of the European Commission, but also at the World Cancer Day, where she repeated the statement that what is really important for Europe is that we need to move to a zero pollution ambition to protect the citizens' health within Europe. Of course, if we think about citizen health, and the recognition that the environment is important, then we also have to ask the question to ourselves, what do we know currently about this environment? And so I always use these pictures of the Global Burden of Disease project, where it basically specifies here the disability adjusted life years, so the life years lost. And basically these ones are the ones that are from modifiable factors from 0 to 100%. What is specified here in these different colors are the different risk factors varying from environment, metabolic factors, and other factors. And basically specified here from 0 to 80 years. And so the take-home message basically from this picture is that on one hand, our field of environmental health and public health have really generated a lot of knowledge that we currently have. And I just put here a red line through there, which basically would argue that about 50% of that modifiable factor that we know is there can be attributed to known risk factors at this moment. But there's also basically this large gray area that we actually see here, which we know are basically modifiable, so there must be environmental causes there. We just don't know what those are, and so that's really the unknowns that we have. Of course, if we think about the knowns, then the question that we have at hand is basically how do we actually move from that knowledge to effective prevention strategies. We know that we have been effective in reducing smoking rates, and that has really reduced the burden of disease. But if we think about obesity, for instance, then effective prevention is still far from actually reaching any goals at this moment. And so we really have to think about design and implementation research, and I think exposome research actually can play a role also in prevention of known factors. The other aspect that we have is basically coming from this gray area, and that is really where we call for new research or new research paradigms, where we really have to think about how do we actually start to gain an understanding of the unknowns. And we have heard this morning already examples of different technology or different techniques that potentially could be used to delve into that gray matter of the unknown in this picture. So again, just to go back to that understanding of the unknown, and this is text from the EU Green Deal and policy briefs, is that the exposome concept actually has been now tightly linked to the European Green Deal effort and the zero pollution effort, where it's recognized by the European Commission that human exposome research has an important part to play in that mission. And I think that is one thing that I think is really important. It's not just now a research activity within the European Committee or European region, but it actually is tied to the larger ambition of the Green Deal. And that also means, on one hand, that there is more funding. On the other hand, and that might be the downside, there's more political pressure to actually deliver with exposome research the goals that they actually have set within the Green Deal. So if we think about the exposome concept, then I basically think that asks for a four-dimensional system understanding. And I view really the exposome concept as a system or a network problem. On the one hand, that is the exposure system, where we have had always a kind of a reductionist view of how we address environmental or other risk factors, and that's by trying to isolate a single factor. Of course, the recognition is now that these actually don't occur in isolation, that there are trade-offs between the different exposures, and they are intimately connected if we think about mixtures and other circumstances. Of course, exposure in itself does not have to be harmful, and so to actually have a disease effect, it does have to basically perturb the biological system, and to actually understand basically those effects, we actually also need to gain a system understanding of biology. Of course, that leads to disease, and again there it's the same thing that we have been focusing perhaps on one exposure, one disease, but we also have to recognize that diseases are also linked. They also come from a system perturbation, and some people might develop one phenotypical expression of that biological change, while somebody else may actually have another expression of a phenotypical change. And so again, we have to gain a system understanding of how those diseases are actually interlinked with each other. I think that's important, and I excuse myself for the fact that I take this example, but in my early life I was a sound and light engineer in the theater, and so if you are a light and a sound engineer, you actually start to recognize that actually by turning one knob, you don't solve the problem of actually getting a good sound. It's actually getting all these different knobs into the right position to actually have an optimal sound. And that's actually the same that we have in effective prevention. It's not changing one single factor, it's actually understanding the system that is behind that exposure, that's behind that effect that we are looking at to actually get to effective prevention. The other, or the last dimension that I think we have to gain a system understanding for is basically the time dimension. And so what I'm showing here is basically a long function trajectory from birth to death, and this comes from a paper of Lange in the New England Journal of Medicine in 2015. But actually this concept that I show you here for long function can actually be used for cognition or for any other function that we have in our body. And that is that at the early age, of course, we start to build up functionality of our organ systems. In this case, this is lung growth. And that exposures in early life can actually impede on basically that development of that organ. And that means that some people won't reach the optimal function of that specific organ system. Of course, when we think about exposures later in life, we all basically have deterioration of our bodily functions after 20. Some of us, like me, are now approaching more the steeper part of that curve. But exposure that happen later life might actually change the rate of decline. And so if we think about early exposures like secondhand smoking, if we think about occupational exposures like silica that actually could have an effect on those trajectories, then there are two ways how you could end up with a phenotypical disease. And that is one where you never reach basically your optimal function and then have a normal decline of that function, and suddenly you basically drop below a functional threshold. Or it is that you did build up your complete function of that organ, but basically because of experience later in life, you basically dip below the functional threshold and develop CPD in this case. And so actually half of the cases are because of accelerated decline, and half of the cases are because people in early life never reach the full potential of the lung function. So I'm showing this that if we actually want to have a complete overview of non-communicable diseases, about chronic diseases, then we cannot just focus on exposures in adult life, but we also actually have to recognize early life exposures, even for diseases that happen later in life. So the European Commission recognized the importance of human exposome research and just launched into 2020 funding for 9 new projects, which involves 24 countries and 126 research groups altogether that are involved in these 9 projects, and a total investment of 106 million euro that went into funding these 9 projects, which are basically depicted here in alphabetical order of the project. What is new is that these projects are not funded as a single entity. They're actually funded as to be a cluster. That also means that there is crosstalk between these different projects to make sure that we actually reach a maximum synergy between these projects. So at the launch event, basically I showed this picture, which was shown this morning as well, that we basically had this investment in the Human Genome Project from the 1990s to 2001 to complete the insights of the human genome. And basically, we have now declared in Europe that 2020 to 2030 is basically the decade of exposome research in Europe. Not to say that we actually will finish it within those 10 years. Saying 10 years has 2 main advantages. One, as a scientist being funded for 5 years, it's always good to say that the program is longer than the 5 years you're funded for, because that means you need additional money later on. From a political standpoint, it's always good to promise something that is not within your 5 years, so you don't have to deliver somebody else maybe at 10 years from now. But this is not a European effort. This is an investment that has been done in the European Union, but basically to solve this picture of the exposome is really a global effort that needs to take place. Now, the Human Genome Project had an approach where each gene was basically parsed out to a certain group, to different continents to actually sequence or to understand that specific part of the chromosome of our genetic material. That is not true for exposome research, and it would also be very difficult to say who is going to do exactly what. But it is important that even with these large sums of money being invested, that in some way we have crosstalk between the different programs that are going on within Europe, within the US, and other parts of the world. So in that sense, I'm very glad that the first step has been taken to start to think about some level of harmonization on some of these untargeted methods that we have heard about, that's happening now within the US, that we heard about in collaboration with some of the European institutions that also within HBM4EU are working on these untargeted emerging chemical screens. So that we actually start to operate not only as a cluster in Europe, but basically as a global cluster to basically develop these new methods. So what I'm going to do in the next few slides is basically try to summarize these 9 projects for you, what they actually are more or less about. This is an incomplete picture because we're just starting, and so I'm not going to claim that I know all the details of all the projects exactly. But if we just think about the first part where we try to understand the exposure networks and systems, then we all take this kind of general approach where we know that human health is really determined by the ecosystems that we actually frequent in. Might that be basically the urban ecosystems, or it might be the rural ecosystems that we have. It's determined by our social environments, by our physical chemical environments, and our lifestyle. Now, if we take these different European projects, then there's a lot of emphasis basically on built environments and basically green space, or basically biodiversity, where I think one of the interesting things is that there is an increased focus on metagenomic research, so understanding basically what the microbes and viruses around our body are doing. There's a large focus basically on chemical exposures, which we have heard in previous talks that is really a very important issue. But I think what is interesting here is that some of the projects are more focusing on environmental exposures, where we now also see that for the first time there's actually quite an emphasis on also the occupational exposures that happen. That's not only from the perspective that was pointed out by Doc Walker, that is a good natural experiment to do some of these experiments, but basically occupation covers a long period of our lives. For most of us, that's basically over 40 years. Exposures that happen are often much higher than what we have in the environment. And of course, the occupation versus environmental and social pressures is one that is actually quite interesting. So we see now E4 and Eximius really focusing more on occupational exposures. I think one of the other interesting things is that we see that the food component, which in the earlier exposome projects was really quite absent in the methodologies that were developed, are now being picked up in these new projects. May that be by actually looking at food contamination, which happens in Expanse, or at dietary components. I think one of the things that is also clear from this picture is that when we think about the social environment, then EcoLife and Hedemet are focusing on that, but it's actually less efforts that are actually put into that domain at this moment, and that might actually be something that we have to see how we can develop that further. When we go to the biological system, and I'm just showing here the usual going from, this is biomonitoring or exposome scans, as we have heard today, the epigenome, metabolome, microbiome, proteome, transcriptome, and the genome. Then we can see, of course, that is covered by the different projects. But if we see now where the emphasis really is, then we see that there's a lot of effort on metabolomics and on epigenomics within the projects, but also the microbiome, which we weren't focusing on in the first exposome projects, is now a topic that is really picked up by several of the consortia as one of the major focus areas. When we think about the different disease endpoints, then this is basically how you can summarize those different projects. On one hand, that is basically focusing on cardiometabolic diseases, that is looking at respiratory diseases, that's looking at the brain, in this case that is mental health and cognition. on immune diseases, and some of the projects that take a more disease-own process where there's not a clear focus on one disease. What is interesting of that is, and I link it back to the first slide that I showed about these different diseases and their genetic components. They're basically shown here that we have this focus here on cardiovascular, lung, obesity-associated diseases, autoimmunity, but interestingly, neurological diseases or neurodegeneration is actually not covered under the current European projects, and so that's actually an area that probably needs further development and emphasis. It's also interesting that the field of cancer research is actually not really covered under these projects. And so I think that's another area where we have to make sure that this area basically also gets covered. So when we actually then think about this fourth dimension and basically time, then we have projects like athletes and Equalife that really focus on early life exposures and early life disease. We have several projects that try to cover the whole life course, and then we have a few other projects that look at adult disease but cover the early life exposures but don't look at early life disease in itself, which is true for Expans and Remedia. And then the projects that focus more on occupational exposures focus really on adult life. So this is basically how you can characterize these different projects that are being funded and which part of the life course they're really covering. Now, the project basically has to lead to sustainable infrastructures. So what we do in the cluster is that the data that we actually generate should go into a general data catalog, which is fair, which means that it's findable, accessible, interoperable, and reusable. So that's going to be done. There is basically efforts on data harmonization so that information between the projects can actually flow. There are tool development basically on the external exposome and on the internal exposome. Developments basically on data analysis and getting new disease associations, which then leads basically to efforts that go into risk prediction and integrated impact assessments, which I will come back to. But in the end, basically has to lead to policy recommendations and suggestions to effective interventions and prevention programs. And so this is really what we want to deliver as the network to the community. I have to apologize, Jelle Vlaanders here in the audience side is actually coordinating the Expans project for me. I didn't realize the screen was this large, but this is Jelle, and I wanted to make sure that you would recognize him. So what I'm going to do is talk a little bit more about the specific project that I'm leading together with Jelle on the urban and chemical exposome and cardiometabolic pulmonary health called Expans. And what I'm going to do is show a few highlights which I think are potentially the areas that I think we should develop within exposome research more. So what we actually do in Expans is we use different designs. And so on one hand, we use studies where in targeted panel studies with few individuals, we have many data elements. And so that could be more than 1 million readouts, either being that on the external exposome or the internal exposome that we actually have on these populations. We combine that with the administrative data sets that we have access to, which allows us to actually get together more than 55 million people health records that we actually can link on an individual level to external exposome measures. And so that basically gives us enormous power to start looking at these different environmental factors, social factors, and also their interrelationships and the interactions that are there. The cohorts that we actually use fill in that space between actually having deep phenotyping and having large millions of people, where the cohorts basically, and this is in Expans more than 10 million people, we have on one hand many data points, different readouts, but not as complete as the specialized studies. On the other hand, they actually have less people involved. And what that actually does is that we actually can start to do triangulation and looking at how well we actually can repeat the readouts that we have on the exposome. Meaning that if we use the 55 million people to actually do a in-depth research for what are the important factors in the environment, we may have basically observations. But of course, those observations may be biased or confounded by things that we actually don't have in these very large administrative data sets. We actually can follow that up in the cohorts that we have, still large cohorts, where we have many more information about lifestyle and other factors to actually start to enrich that inference that we have. And from that, we actually can go to the biological system by actually going to these panel systems to figure out how that actually works on a biological level. The other way around would also work, where you may have observations that you do here, but actually power-wise may be underpowered, and we actually can link that back to see what the impact is actually on the full population of Europe. And so I think this is a very powerful approach where we actually start leveraging off the information and by standardizing the information between the administrative cohorts, the deep phenotyping, and the cohorts that we have, we actually can play around and see what actually lines up in the inference. The other aspect that we are really focusing on is basically the use of high-resolution mass spectrometry. We have heard this before. On one hand, that is basically using metabolomics platforms, which really gives us a good coverage of all the biological pathways that are important for human health. But on the other hand, we have a large investment in actually doing untargeted chemical screening by using the platforms that actually are developed here in Mount Sinai and at Emory University to actually get wide coverage of the environments, the so-called exposome scans. So that focusing on chemicals is important, is something that we recently laid out in Science. By arguing that the chemical world is very complex and vast. We have heard that we have many chemicals on the market, but if we actually look at those chemicals on the market, most of them actually have not gone through rigorous testing as we actually would mandate today. So there's a legacy that we actually have. The other aspect that we have, and I always call this the same as in big data, we have a large variety of chemicals shown here as well, and Doc Walker showed this as well, with over 70,000 chemicals within the blood exposome database. And if we actually go to information about PubChem, then actually that becomes much larger. There is also a large increase in volume that basically also was shown before. But what we'll also have is that there is a high veracity. We have chemicals coming on the market, and before we start building up an evidence base, that actually might be superseded by another chemical. So if we think about bisphenol A, now we have bisphenol F, and to actually build up the evidence base for one chemical before we actually have an idea of what's happening, we're actually 10 chemicals further. And so what we argued together with Gary Miller in the Science paper is that we have to move away, and we have to understand biology so that we actually can not just look at one chemical at a time, but basically treat it as a system. We have seen this before, and this is a slide board from Doc, that if we actually want to address this chemical complexity, then we have to move to more untargeted and wide screening of chemicals. That has been laid out before. One of the issues that we have when we do that is annotation. Annotation of our biological system is much better than the annotation of chemicals when we do these high-resolution mass spectrometry. We have a poster tomorrow of Jeroen Meijer, who unfortunately could not attend because of the coronavirus, but what he has done is basically aggregate all the information from emerging chemicals in collaboration with the Norman database, basically made that into a mass spec ready database. We simulated the metabolites by using biotransformer and basically also have linked it to prediction of both toxicity and environmental fate. So the idea that Doc basically presented of it would be good if we actually have an idea about how present or how common these exposures are. That's now done within this database, which is now implemented in the 5 labs in HBM4EU that do chemical emerging screening using the same annotation database. And we hope that that annotation database will be available for the wider community within the next weeks. That this approach can be very successful was already laid out by Doug by showing the trichloroethylene example, which was the first example where we started to believe that we actually may be able to actually harness this information in a useful and meaningful manner. I'm showing you here another example that is part of the Exposomics project, which preceded the current Exposome project, where we basically used incident cases of asthma, adult onset asthma, and cardiovascular disease. We basically selected about 300 cases and 300 controls by disease entity, ran metabolomics on the prospective samples, did a metabolomic-wide association study by first looking at how is that related to the disease, which is basically this line where those signals were taken, pathways were calculated and looked for which ones were enriched. We did the same by then basically looking at the intersection of air pollution on the metabolome as well, and basically then came to the conclusion that the linolate pathway is really important because that's where we see actually mediation between air pollution and the adult onset asthma. And actually the same observation was done for cardiovascular disease, that we now appreciate that the same biological pathways might be underlying. Just going to skip that. So the other area that we're really investing in, and I think the next talk when we basically talk about geospatial modeling and using satellites, is that we're investing in actually building up this multi-layered information about our environment. So that's basically air pollution, greenness, walkability, cyclability, and all the other factors. But of course, that doesn't lead to individual exposure. So what we actually have to start doing is basically take this field-based modeling, which is our GS-based modeling, with agent-based modeling, which is much more common in the social sciences, where we start to model human behavior and how they actually interact with their environment. By combining those together, we actually can go to much more individualized exposures. This is something that we're doing together with Google, and so this is called the Google AirView project. I'm showing here the results that Joshua Apte and others obtained for LA and for Berkeley. But this basically starts giving us very rich hyperlocal information by putting sensors on mobile platforms. And together with satellite information, which actually have a better temporal coverage, but have a less grained spatial coverage, by actually merging and assimilating those two data sets, we actually can come up with much better information than before. Of course, being Dutch, we don't believe in cars, so we actually now have fitted bikes with sensors, and so we have over 500 bikes in the province, and so really involving citizens in collecting data, understanding and interpreting data, which I think is one of the important things that we do. What you see here is now how we actually have been able to map basically large parts of the province by actually building these into bikes of citizens. That these kind of information can really be very helpful is actually shown in this slide, where in the Google Street View cars, we actually have been able to put in ultra-fine particulate monitors, really high-grade equipment. And one of the things is that although we regulate air pollution en masse, given that ultra-fines basically don't contribute to mass, but are very important in particle numbers, they're actually not necessarily covered by the current regulation and the ambitions that we have. So we were the first to basically link this to the incidence of cardiovascular disease by actually having full geospatial maps. And what you can see here, and this is just the first analysis that 2 of my PhD students did, is that what we can see is that ultra-fines in this particular study seems to be driving the risk of having a cardiovascular event much stronger than what we see with PM2.5 and NO2. And so this is an important insight that we may gain by actually having these different sensors. I'm just going to skip this for time. I just want to put out a cautionary tale as well, and that is that, I mean, I'm showing you here very much exciting things that one can do, but I just wanted to warn for optimistic linkages. So a lot of studies have recently been published on light at night, taking satellite imagery or pictures of the International Space Station and linking that to the occurrence of health. We actually did a study where we put people a light sensor to see what their individual light experience was also at night. And you may appreciate these very high correlations, which are virtually zero. There is no correlation between light at night and actually the light circumstances that people have in their bedroom, and that's simply because we have curtains and other things that we do to avoid basically light entering into the room. That seems very obvious, but one of the things is that light at night is very highly correlated to air pollution, 0.7, to urbanicity, 0.7. And so probably what we actually are looking at in these studies is not light at night, but it's some level of urbanicity or other risk factors that are related to health. So we have to be careful in optimistic linkages. The last thing, and then I'm going to conclude, is that the other thing that we are investing in is actually getting this system overview of the biological system. And one of the areas that we are making progress is this idea of building so-called biological scores, or basically this allostatic load scores, and this is work by Mark Shadow-Guillaume at Imperial College, where by using UK Biobank and all the biomarkers that have been measured in UK Biobank, we actually have constructed for each organ system the functionality score for each individual, summarize that into a biological health score, and then ask the question if that is actually perturbed, are you at increased risk of developing disease later in life? And that's true for cancer and for cardiovascular disease. And so when we actually start to have more of this biological information, then we actually can start to develop these into what we call exposome risk scores, to actually start to know which environmental components are important, which biological systems are important, and actually can we actually get that into one metric. So this is my slide and combination of Gary Miller's slide that he showed in Brussels. And so I think really the full potential we have is not by actually contrasting the human genome versus the exposome. Actually, they go hand in hand, but we actually have to have a comparative good overview of both aspects to really get to the true nature of health and disease. So with that, I just wanted to acknowledge my group at the Utrecht University, the people at Imperial College, especially Mark Shadow-Guillaume, and the three musketeers, how I would like to call them, Doug, Gary, and Dean, here on the US side, which have been imperative for many of the research projects that I have done. So for that, I just want to thank you, and I'm happy to take any questions.